Hi, I'm Harsh. I'm a PhD student in the labs of Jane Carlton and Rich Bonneau at NYU and Johns Hopkins. So proteins exhibit a spectrum of intrinsic disorder. So for example, what I'm showing here is an ordered protein in which we have a very strong understanding of the function and structure relationship that's found within these sorts of proteins. But intrinsic disorder is found as regions of intrinsic disorder, so intrinsically disordered regions, or for proteins that are predicted to be or experimentally verified to be entirely disordered, so intrinsically disordered proteins. And this is an emerging structure-function relationship, and that's partly been limited by the experimental characterization of IDRs and IDPs being really costly and low throughput. And so we've turned as a field to rely on accurate prediction and validation of disordered residues, but though this remains kind of challenging. Most of these function prediction models for intrinsic disorder rely on foundational protein language models. So in this table, I'm showing encoder-only protein language models and the large data sets that they're trained on, such as UniRef, Magnify, AlphaFold database. And the reason why these foundational protein language models are suitable for this task is because they rely on training on this large corpus of sequences, which then allows them to produce embeddings that encode evolutionary, structure, sequence, and function signal, which are useful for downstream tasks such as structure prediction, protein design, function prediction, and mutation effect prediction. However, when we look at how these foundational protein language models are trained, we see that they're trained using a mass language modeling task, in which an input amino acid sequence is tokenized and certain tokens are masked out. And the goal of the model is to predict amino acids at masked positions given the context of unmasked amino acids. This has a limitation because MLM alone doesn't learn the specialized biological signals that are annotated in databases such as UniProt, which contain troves of information about protein sequences. And so what we're hypothesizing is a multitask fine-tuning framework that can utilize these deposited annotations and infuse the latent embedding space with this information. Previous work in this field done by the Ross Lab shows that fine-tuned protein language models that incorporate this biological signal in the embedding space has improved performance across a variety of tasks seen here on the x-axis, across a variety of protein language models seen on the y-axis. And specifically, when we look at intrinsic disorder prediction, we see that the fine-tuned variants, which have incorporated the biological signal of intrinsic disorder into the embedding space, outperform all of the non-fine-tuned variants. But furthermore, when we look into how these fine-tuned protein language models arise, it's through two techniques. One, using either an annotation vector, or two, using a feature matrix. And here an annotation vector could be representative of any biological signal, represented as a continuous vector or as a one-hot vector. And then you have your feature matrix, which consists of residues by annotations. And the model now is tasked with learning all of the implicit high-dimensional interactions between the sequence and each of your annotation channels. And so we propose using what are called feature-aware tokens, which encode annotation patterns as a linguistic unit, similar to tokenizers, and therefore can capture the patterns of annotations that are found at residues within proteins. To this end, we build an end-to-end framework for prediction of disordered residues and their associated functions. And so the first part of this pipeline is distoken, which are disordered tokens. We then use those tokens to fine-tune a protein language model, and then we test the performance of these embeddings using very simple classifiers downstream. So we can get started with distoken. So distoken was trained using 47,000 sequences from the homology merge dataset that's found within MobiDB, and this comprises about 21 million residues. Particularly noteworthy is the aggregated representation of both the feature matrix and the residue embedding prior to using the encoder, the quantization step, and the decoder. The task of when we're training this model is to reconstruct the feature matrix in the initial input. And during inference, what we can do is we can freeze all of the weights and use only the encoder and the quantization step to produce for each amino acid a discrete distoken that corresponds to it, and therefore building a disorder-aware vocabulary. When we take our 47,000 sequences and tokenize them, we can see which features were activated for which individual tokens. And so what we'd want to see here is specific DIS tokens being enriched with specific features, which therefore means that it's capturing the features, the annotation patterns that are found within that residue every time it's annotated. Here, DIS token 4, 5, and 0, for example, were tokens that were assigned every time the disorder annotation was present. So here being homology disorder merge. And we could also see that DIS token 0, for example, is an aggregation of most of the predicted annotations that are found deposited within MobyDB. Looking at the distribution of DIS tokens across the 47,000 sequences, we see that DIS token 1 has the most number of assignments, and that intrinsically makes sense because of most of these residues within the dataset being found within ordered regions and also within domains. And furthermore, when we take the latent embeddings of the codebook that we'd used to train DIS token and we analyze the mutual information between each of those codebook centers, we see that there's a natural pattern of discrete and shared features that are used within each of these codebook centers. We wanted to next evaluate whether DIS tokens were assigned in contiguous stretches. And so what I'm showing here is an example of two proteins that were randomly drawn from our data set, in which what we first noticed is that DIS tokens were assigned in contiguous stretches. We next noticed that the specific DIS tokens were correlated to regions of consensus disorder, here denoted in gray, or linear interacting peptides, here denoted in yellow. And when we visualize our DIS tokens on the AlphaFold structure, we see that they correspond, the DIS tokens that are associated with disorder correspond to regions of low AlphaFold PLDDT. To be a little bit more concrete with our analysis, we wanted to quantify whether tokens were contiguous. We looked at the entropy within disordered and ordered regions of proteins to see whether the codebook was being utilized to a greater or less extent between these two regions. And we find that ordered regions have higher entropy, meaning that they're utilizing more of the codebook, versus disordered regions have a lower entropy. And we also benchmark this against 3DI, which is a structure-aware alphabet and another tokenization approach in which the 3DI tokens we hypothesize were capture disorder or the absence of order in the structural alphabet. And what we notice here is a much higher entropy for DIS tokens for disordered regions and also for the ordered regions, kind of also highlighting the complementarity approaches between both of these tokenization schemes. When we're looking at the average, another way to quantify this would be through the average autocorrelation, in which across a variety of different lag values, we see that DIS token is more assigning more contiguous stretches of tokens than the 3DI across the entire protein sequence. And so taken collectively, these results suggest that DIS tokens are contiguous and they also have low entropy. We can move on to the fine-tuned protein language model that we used that was fine-tuned using these DIS tokens. And the way that we went about this was actually keeping the original language model head and adding an auxiliary head next to the language model head to train on. And so the model is trained using a combined loss of both its original task and also our DIS tokens. And in experiments, we found that retaining the original task was informative for the model to not catastrophically forget the original embedding space that it was trained on. We wanted to be able to visualize these proteins. We wanted to be able to visualize our embeddings and how we've shifted the embedding space once we fine-tuned. And so here, what I'm showing are 4 different variants of the model that we fine-tuned. Here is ESM binary, which is just using a binary vector of order or disorder. And then we have ESM distalk, which is using our DIS tokens, ESM 3DI, which is using 3DI tokens, and the complementary approach, the combined of both disorder and ordered associated tokens being distalk and 3DI. And what we see is that both the base model has a diffuse representation of intrinsic disorder from the sequences that we ran this plot on, with being sequences from the DISprot 9.3 dataset, in comparison to ESM binary and ESM distalk, which show that disorder has been encoded almost as a separate arm within the embedding space. We see similar results for 3DI in the combined model, but it's a little bit more diffuse than what we see with distalk, which was an expected finding. Another way to be able to quantify our embedding space would be to dimensionally reduce the embeddings down to a PCA plot and measure the separation of centroids from proteins that were mostly ordered and mostly disordered. And what we see here is that the model ESM disttalk and both ESM binary are able to separate out ordered and disordered proteins to a greater extent. However, from the previous results, you'd ask, why not just use the ESM binary model? And the reason why you'd want to use the feature-aware tokenization approach is that the embedding after the model has been fine-tuned remains close, contains high cosine similarity to the original embedding space, which suggests that the model hasn't forgotten what the original task was. And furthermore, when we can quantify this by using the model's fine-tuned representations on a logistic regression task on the secondary structure data set to analyze whether the performance of the model differed from the base representation here shown as a horizontal line, our fine-tuned variant shown here in the bars, and then the binary shown here, which means that the binary representations after the ESM binary representations post fine-tuning are a little bit more different than the base representations, which means you wouldn't be able to use them in other downstream tasks. Lastly, we wanted to train a classifiers built on top of our frozen ESM disttalk embeddings, and we wanted to keep these classifiers minimal on purpose. And so we simply just ran a convolutional neural network over the per residue embeddings from the fine-tuned protein language model to predict intrinsic disorder, binding, and flexible regions. We trained this model using a sequence similarity split that was produced by Pang et al. What we find is that when we're comparing all of our fine-tuned models here, ESM disttalk plus CNN shown in the black lines, we see that there is a boost in performance, particularly for IDRs jumping from 0.85 to about 0.94. And same thing for the same pattern being seen for binding and also for disordered flexible linkers, so DFL. And we see that models that were using the 3DI tokens or the combined dist token plus 3DI lag behind, confirming that our disorder-aware codebook is yielding better downstream features for than pure structure quantization approaches. And all of this is without touching the base transformer for the ESM disttalk. So this was just a downstream classification task. And we see a performance improvement for these fine-tuned embeddings. Furthermore, we wanted to be able to quantify these. We wanted to be able to quantify the results and the performance of the model on K2 benchmark data sets. And so when comparing against 65 published models, we see that the ESM disttalk plus CNN model achieves a rank of 1 across 3 data sets and a rank of 2 in 1. And what's particularly surprising here is that these were all using really minimal classifiers that were trained with a sequence-only input, which is a testament to how effective the fine-tuned representations are. So in summary, we developed a disorder-aware vocabulary, dist tokens, for use in fine-tuning protein language models with intrinsic disorder features from MobiDB. We fine-tuned an existing low-parameter ESM2 model with dist tokens, ESM disttalk, incorporating explicit disorder-aware information into the learned per residue embedding. We trained these really small minimal classifiers on top of these frozen embeddings that achieve really good performance, outperforming both the baseline models and published K2 predictors. And for next steps, we were particularly interested in deploying our dist tokens for homology search, also to create a phylogenetically informed disorder residue prediction model, and then incorporating other residue level features and protein level features within our tokenization approach to build a more robust feature-aware token. All of this work wouldn't be possible without my acknowledgement, specifically Dan, Omar, and Nathan, who've been essential to this project, but also both of my PIs, Jane and Rich. With that, I can take any questions.
Okay, thank you very much, Harsh. Questions?
Yeah, it was really interesting. I have kind of a practical question, and I don't really know about the tokens here. Are these like categorical tokens? Do they come out as, you represent them as characters, or are they kind of vectors of numbers that end up representing amino acids under different disorder conditions?
So you could do both ways. So the tokens can be represented based off of their learned codebook embedding, but also just the character, like discrete class 1 token that's used in the fine-tuning approach. And we actually did 2 different versions of it, and we found similar performance. So simplicity is easier, so we decided to just use the character representations.
Hi, great talk. I may have missed it. When you reported the accuracy of 80 to 94%, was that for disordered residue prediction?
Yeah.
Okay, great. So there's been work that there's a maximum capacity for disordered residue prediction that could be around 85%. And 94 is much higher than that. But that was estimated based on similar or same sequences that have different structures in PDB. One is ordered and one is disordered, depending on the experimental conditions of crystallization.
Yeah.
And so do you capture some form of crystallization parameters when you are going above the maximum accuracy, or what is going on there?
Implicitly, I would think so, because the data set that we used to train this token was from the homology disorder merge data set, which contained the 47K sequences. And I presume that in those sequences, since it's a homology related task to identify disorder, you would be capturing some of that information. So maybe implicitly, but definitely something to think kind of more concretely about downstream.
I'm not sure exactly how you generated the tokens, but there may be, there seems to be a little bit of circularity in that the tokens relate to disorder, and then you're trying to predict disorder using the resulting embedding.
Yeah, I can see that. So each of these steps are discrete, and they don't rely on, there's no shared weights between the tokenization approach and the fine-tuning approach. Furthermore, the data sets that we used to evaluate the model were split based off of sequence similarity to the training data set. So we were trying to be as agnostic as possible.
So I might miss that. Do you have any pre-trained models that you start with? Or if you do you do any optimization there?
So just for proof of principle, we used a really low parameter ESM2 model because we didn't really see a reason to go beyond 150 million parameters. And we fine-tuned all of the weights within the model. So we didn't use LoRa or anything.
I have a quick question. So this was trained on regularly sized proteins, say 100 and more.
Yeah.
And once you get down to a certain length, everything gets disordered.
Yeah.
How short is the shortest sequence I can put in for these things?
So 128 is the shortest, and then the longest was 1024 because that's the limit for the model, for the downstream protein language model. We're kind of also
interesting choice of numbers. Powers of two.
Yeah, we're also working. There's a reference. We're also working on trying to just create a fine-tuned version of the model in which you could predict the disk tokens without relying on the vector quantized variational autoencoder.
Hi, thank you very much for the talk. You mentioned that you used a homology-based module from OBDB to you also mentioned that one issue with intrinsically disordered regions is they are hard to characterize and they remain quite uncharacterized. So how do you tackle this problem of detecting homology if there's little experimentally characterized in the first?
Yeah, that's a great point. So for the disorder homology merge data set, it's like one of the curated data sets that's found on MobiDB. We wanted to use that data set explicitly because if we want to use this model downstream for homology-based search of disordered proteins, we can implicitly learn the relationships between disorder and homology, and then therefore kind of expand to more mutated sequences that have disordered. Yeah.
Okay. Any other question? Okay, one more question, and then we have to move on to the next speaker. The next speaker is here. Please start setting up.
Nice talk. So in identifying the disordered region, do you also end up in identifying SLIMs?
Yes, it is one of the. Do we incorporate SLIMs within the data set? Yeah. Not explicitly, just LIPs, so linear interacting peptides, which should include SLIMs as a subset based off of the annotation tree.
But do you get as an output also predicting pick slim images from this?
We could, yeah, we could.
Okay, our next speaker is Catherine. Are you in the room?